Good morning, everyone.
Good morning.
And welcome to our winter-spring Edward A. Ames Seminar series. I see a lot of familiar faces, and I see some new faces, which is really terrific. I want to welcome everybody on the webinar. Today, we're kind of thrilled to have typically, we'd like to have folks that are either doing work directly in the Hudson and the harbor, or funded by the Hudson River Foundation. Today, this something a little bit different. We're bringing somebody that is doing some work down in the Chesapeake that we think probably has some relevance here. That some of you in the audience has heard Dr. Goodwin's talk 2 months ago on a webinar down in Washington. And Jim Lodge, our oyster aficionado researcher here, said we probably ought to have this guy up here because he's got a lot of really interesting things to talk about. I wanted to spend 1 minute, those of you that don't know what we are up to here at the Hudson River Foundation, as to why we would have invited somebody from Chesapeake to talk about oysters. We got involved with oyster research a few years ago, which came out of some work that we were doing with the Corps of Engineers and the Harbor and Estuary Program, and trying to develop some scientific underpinnings for restoration in the harbor. And we, along with some other folks at Cornell, wrote some target ecosystem characteristics to find 12 now, and sort of handed them off to the Corps, and they form the basis for the Corps' Comprehensive Restoration Program. And one of those TECs has to do with oyster restoration. And if we're going to move toward oyster restoration in this region, there's a fair amount of things that we don't know about oysters and how they survive in this environment. And we embark on a journey with some of you in the room to create some pilot projects and other forms of research to get to the point where we can make some decisions as to whether oyster restoration would be feasible. We're not there yet. Jim is our point person. You know Jim. He's been working, and he's got gained a lot of knowledge, and he's become very frustrated with this topic, but he's still moving ahead. So we're pleased to have Jake here. Dr. Jake Goodwin, who got his PhD at the University of Maryland, and now is a postdoc at Rutgers, but I understand he spends very little time in New Jersey because he's still busy down in Horn Point and Woods Hole and doing a whole lot of really fascinating things with studying and identifying larval oysters. So we're thrilled that you're able to come. And by the way, happy St. Patrick's Day to everyone. And we're glad to have you all here on such a momentous occasion. And hopefully the traffic wasn't too bad for you to get in. A little bit of housekeeping. We asked everyone to try and sign up ahead of time, just so we make sure we have a seat for you, and then we can get in contact with you. We do have a sign-in sheet that if you didn't register with us, that we can at least get your address, your email address, or phone number, or another address that we can stay in contact with you. We're pleased to have a number of people on the webinar, and they can hear us, but they can't talk back. They can post some questions later on, and we will ask the folks on the webinar that I'm sure you probably have been instructed by Melissa. But at the end of the talk, you have an opportunity to write in questions, and Jake will do his best to answer them. So without take care of all the business? Yeah. Okay. Jake.
Thank you. Thank you to the Hudson River Foundation for inviting me here. Thank you all for coming, looking into this research. I think the 2 case studies I'm going to talk about today do have some merit in New York as well. There's other places that oyster restoration is taking place. I'd also like to thank my supervisor, Dr. Rich Lutz. He allows me the freedom to sort of go off on my own and explore ideas. And all of this work was done in the University of Maryland at Horn Point Laboratory. So oysters are important. We probably all know that. They help form habitat for other species, they're also ecologically important. They can filter algae out of the water, and they're commercially important. Although here, your purposes may not be to harvest the oysters in certain areas. There's several species of bivalves that are, as you all know, are commercially important around the world. And understanding the biology and ecology of oysters and other bivalves in particular can help us improve restoration strategies. Here's a slide of history. Back in Chesapeake Bay, and probably New York Harbor as well, there were so many oysters that they would actually block navigation for ships. The population in Chesapeake Bay is less than 1% of historical population. So here's a plot that a lot of people that work on oysters in Maryland will show. And it's the decline due to harvest over the years on the x-axis. And this is millions of bushels. So there was a huge decline and then a later decline later on. And the biggest decline was due to overfishing, which happened all over the East Coast. So 125 years ago, we had 12 million bushels, 26,000 watermen, 17,000 boats were working. We had 330 acres in Chesapeake Bay of oyster reefs. And after the habitat destruction in the 1920s, we ended up with 2.5 million bushels and only 200,000 acres of oyster habitat. And there was a further decline with disease, Pipinthus marinus, causes a disease called derma in oysters, and that knocked the populations down further. And that also affected oysters in Delaware Bay as well. So the main goals of oyster restoration, at least in the Chesapeake and probably in other places, are to increase the adult numbers. So we want to get more. We actually want to get more adult oysters out there, but we also have a goal to not just plant oysters in certain areas, but set up big sanctuaries. So these sanctuaries can create larvae that will go to other areas and create more habitat. So. And eventually the goal is to have a self-sustaining fishery. Right now in Chesapeake Bay, we plant spat on shell in different areas. So that's the state pays to put spat on shell out in these different restoration areas. So the goal is to not have to do that anymore, everywhere on the East Coast. So science can help us with these restoration goals, and I'm going to talk about that today. So by studying oysters that are out there, we can help increase the adult numbers and utilize and see what's utilized by other reef organisms. And understanding the larval stage is really important because this allows you to understand sink and source dynamics. So what reefs that you put out will contribute larvae to go to other areas, and where will those larvae go? So this is something that we're trying to figure out. So as I said, at Horn Point Hatchery, they put spat on shell out. So they rear the larvae in tanks. The larvae set on shell, and then a boat actually takes the spat set on spat as juvenile oyster. It takes the shell out to different areas in the Chesapeake Bay to be planted. Some other efforts are being done, though, also by the Chesapeake Bay Foundation. That's a nonprofit organization in Chesapeake Bay, and they're putting out structures called reef balls. And right now, the effectiveness of all of the programs are being monitored by Ken Painter's lab at the University of Maryland. So reef balls. Reef balls are these three-dimensional concrete structures, and they, in Chesapeake Bay at least, they pre-set the spat onto the reef ball before they plant the reef ball out. But in high salinity areas, people will just put the reef ball out, like in Virginia, and they'll get a natural recruitment on that. But so Chesapeake Bay program has been using these reef balls essentially because they deter poaching. So it's sort of an off-the-record fact that you can't run your dredge through a sanctuary that's got a bunch of these structures because you'll ruin your equipment. But no one ever studied how well oysters really grow on reef balls. There's nothing in the literature. And if anyone knows of anything in the literature, let me know because I'm writing a paper right now. How big are those? Oh, I'm sorry. Yeah. So reef balls take about a square meter in terms of diameter, and they range in height, but they're less than a meter in height. So they're not big navigation hazards in terms of, unless you're in a really shallow area, really shallow, less than a meter. So just the basic question is, do reef balls enhance oyster populations, and do they support biodiversity or reef-associated fauna? There's new studies that show that reef-associated fauna can help with denitrification. So that gives an enhanced effect to help cleaning the water and taking nutrients out of it. And that's a study by Lisa Kellogg. So we wanted to go out and determine the abundance and size classes of oysters on reef balls, and then assess this biodiversity and the abundance of reef-associated organisms that were on the reef balls as well. Then we got to take this nice boat out to do it. That's a picture of an oyster reef in Chesapeake Bay. And the site was Cook Point, and it's right there. This is the Chesapeake River. So let's see, Annapolis is over here, and the Bay Bridge just goes over here, and that's Chesapeake.
Excuse me, are you going to discuss the transport of sediments interfering with reef balls?
No, I didn't look at sedimentation, but what I did was compare the traditional spat on shell methods with reef balls and look at how oysters grow on both. So this messy diagram just shows you that there's a lot of work that's been done in this location. So everywhere that you see a line, a colored line, is where they planted spat on shell. It's where a boat drove around and actually planted, or they literally hosed off this shell with spat on it all over this area. And then all the dots represent 3 reef balls that were placed in those areas. So a lot of work's been done at Cook Point. So I found 2 sites, one that I call the north site and one the south site. And I was able to set up an experiment with reef balls, spat on shell, and then areas just sort of away from the reef balls. I wanted to look at oysters that were right next to the reef balls as well, to see if they had some sort of protection from predation or other effects, to see if there was a difference. So we had divers go down to these areas. Okay, so this is the south site. And a really important note is that at the south site, all of the treatments were planted in 4 years prior to looking at them. So the oysters on the reef balls were the same ages as the oysters in the control and the other treatment. So here's a schematic. That's not the Chesapeake Bay. That's down in Florida. So they use reef balls for sponges and corals as well. But they're about 1 meter in diameter, and some of them are less, or half a meter in height. But the surface area, the total surface area taking out these holes is 2.79 meters cubed. So there's 2 ways to look at and compare reef balls to other restoration sites. You can look at it from just 1 meter squared, where you just take a quadrat. The diver will just take oysters that are in a quadrat, and you compare what's in that quadrat to what's in the quadrat on the spat on shell. Or you can, that's one way to look at it, which we did, and we also looked at it as 1 meter squared of restored bottom area. So when you put a reef ball out, you're restoring that whole area. So you have to multiply that by the entire surface area, which ends up being 2.79. So we analyzed this 2 different ways.
You put the spat on shell down, was on top of a bed of shell to begin with, or was it sandy bottom?
It wasn't sandy. There was some shell there, but there were no oysters there. So there were no previous oysters. They were restoring the site. I mean, there could be natural spats out there that they didn't know about. But I'll show the size distributions. I can touch on that. So divers went down and collected 15 quadrats from each of the treatments. So again, there was a control treatment that was away from the reef balls that spat on shell. There was a treatment that spat on shell near the reef balls. And then there was the oysters actually on the reef balls.
Sorry, the planted density of the spat on shell, do you have an estimate?
So we don't have the initial densities. So that's one of the, so they already had planted these. No one's ever looked at this. So I didn't get to set up a controlled experiment. I had to go find areas to look at in that crazy track line that were comparable. So it was really hard just to find a study site. That's the next step. Yeah. So keep in mind, I'm not trying to say that oysters growing on reef balls are better than spat on shell. I just looked at the 2 planting methods in that area. So in the end, I mean, the conclusion is that oysters grow well on reef balls. That's a general idea. And I won't spoil the reef-associated fauna yet. So we did a condition index, so that's sort of a way to tell the health of the oyster by tissue weight, dry shell, dry tissue. And there was no difference with oysters on the reef balls versus oysters on spat on shell. There was also no difference in Perkensis marinus infections. So here's abundance, and remember that we didn't have initial abundances. So, but even without the initial abundances, oysters still appear to grow quite well on reef balls after 4 years. And here's a size distribution. So there's a wider distribution of oyster sizes on the reef balls. So after 4 years, you don't really expect to see oysters that are less than, say, 50 millimeters. So this means that we're getting some natural recruitment on the reef balls. And we got some natural recruitment on other areas, but not quite as much. So this is the abundance of just size classes of oysters. And I did it because market size is around 76 millimeters. So I broke it up into 3 size classes. And these oysters over here are likely new recruits. And again, there's statistically significantly more on the reef balls. So after all this time, the reef balls are getting natural recruitment, and the oysters that were planted on them are survived. That's what this means. So we did a Shannon-Weiner diversity index, and just to look at different organisms. So these are the organisms that stood out. There were other organisms on these plantings, but mussels, polychaetes, barnacles, anemones were all statistically significantly higher on the reef balls than they were on the near reef ball or control treatments. The actual index of biodiversity wasn't that different between the treatments because we have sort of the same organisms, but there's a lot more of these other organisms on reef balls. And these mussels also help with denitrification. If you look at Kellogg et al. 2013, and you can see the reef balls are recruiting these other bivalves. So they're very good at recruiting. And it could be the hydrodynamics. I'm not quite sure why the reef balls recruited these other bivalves better, but they did. So from that study in conclusion, oysters were more abundant on reef balls than they were on the spat and shell treatments. And it's likely due to the three-dimensional structure. But oysters are surviving over time on those reef balls. And there was some natural recruitment. The biodiversity was slightly higher on the reef balls. But all these organisms, the mussels, the polychaetes, anemones, were significantly higher in abundance on the reef balls compared to the other treatments. So the conclusion to take home from this is that evidence suggests reef balls can promote oyster populations and enhance reef-associated fauna. And I'm working on a paper right now with Elizabeth North that's going to be way more in-depth than what I just showed you. That'll also show the just direct surface-to-surface comparisons. And a lot of these conclusions cross over, and then it incorporates the other side as well. So hopefully that comes out in the next couple months, right? So with this case study, looking at oyster reefs and looking at structures for adult oysters, we can help with our goals of increasing adult populations, increasing the habitat used by other organisms, and enhancing this habitat value to try to get to work towards a self-sustaining fishery. So this is just reiterating that we're looking at a case study with the adults, and now we're going to jump over to the larval side. And the effectiveness of reef sanctuaries depends on the transport of this larval stage. There's a lot of unknowns with trying to study bivalve larvae in general. In science, we've been trying to understand how to identify them for about 400 years we've been studying oysters and still didn't have a fast way to identify them. And my supervisor, Rich Lutz, back in the 80s, he had scanning electron microscopy, which was the new emerging technique, and it's still the best way to identify them, but it takes a lot of time. So to identify a sample of larvae might take a week or more with that method. So that's the biggest challenge, is just trying to identify the larvae. But if you can start to go out and sample the larvae, you start to understand things like behavior and mortality. So oysters, when they reproduce, have a larval stage that's transient in the water column. So they're part of the plankton. And they cannot govern their position vertically, or horizontally, against the current, but they can govern their position vertically. So if a larva is at the top and you have different kinds of flow patterns in your system, it'll travel one way versus another. So, and understanding mortality can also help us. And these things we can put mortality calculations and behavior of larvae into particle tracking models. And then we can use these models to try to figure out where oyster reefs will go, sources and sinks for the different reefs and sanctuaries that we put out in different waterways. So again, back in the Chesapeake, same system, the Chesapeake River, that's where I did this work. And the main objectives were first to create a visual guide of the bivalve larvae in the Chesapeake River to enhance the identification of oyster larvae. And then test and improve Shelby, which is a pattern recognition software that I'm going to talk about a little more. That's just what we call it. And then we had to develop a way to do it fast, so a benchtop automated image acquisition system. And then, of course, finally, after getting all that done, we went out in the field and were able to collect samples and identify oyster larvae and get at the behavior and the mortality rates. So if you look at a sample of seawater from the Chesapeake Bay, on our microscope, it looks like this. And as I said, it's very difficult to identify oyster larvae. And there are some molecular techniques like quantitative PCR or real-time PCR, and fluorescence in situ hybridization. But these are expensive and time-consuming, and they are also susceptible to errors. So the shells of bivalve larvae form a unique pattern on polarized light because they birefringe, so they refract light in two directions. And if you look at that same sample under polarized light with a wave compensation plate or a lambda plate, lambda made out of gypsum, then all of a sudden the bivalve larvae, because of the calcium carbonate, illuminates. So this is the exact same photo. It's just one of this image on the bottom has polarized light with full wave compensation plate. So we call it Shelby, and it's for shell birefringence, and it's an image analysis technique. It was first used by Scott Gallagher, whom I'm working with at Woods Hole, and Christine Thompson was his student. But basically, up there, they figured out that it works, and you can tell the species apart. But they were using hatchery-reared species to try and identify field larvae, and there were some issues with that I'll go into. So for us, we spawned, reared, and created a visual library of the 8 species of bivalves that spawn at the same time that oysters do in the Chesapeake Bay. And so we have 1 oyster larvae species. We have 5 different clam species, and then there's 2 mussel species here. So this is coming out in April edition of JSR, the Journal of Shellfish Research. And the good thing about this is when we create our categories or training sets, which I'm going to define in a minute, there's only 1 oyster, so the pattern is very distinguishable from these other species. If you had a system, say, in the Caribbean, where you had a lot of different oyster species and you had lots of clams, if the patterns are not distinguishable, the software will have a harder time. So this whole technique may only work in an area where you can basically visually define your target organism, and that it doesn't look the same as something else. And you can see some of these clam patterns look very similar. And the patterns change throughout development. So you have to create quite a big library. So even after you can do that, though, and identify these oyster larvae in the Chesapeake, this still takes a long time. You still look under the microscope, you still have to count these to try to figure out how many were in a sample. Say 200 liters of seawater. It could take you a week still. So the next thing we wanted to do was make this a lot faster. So we wanted to test and improve this Shelby. Oh, I'm sorry, I jumped ahead. So I said that at Woods Hole they had used this before, but they used hatchery-reared larvae to try to identify field larvae, and there were issues with that. So we actually, first we wanted to make sure Shelby worked in the Chesapeake Bay at all. So it uses this pattern recognition software, kind of like the FBI does for irises. It's a support vector machine. That's really what it uses to classify things. And they showed, Scott Gallagher and Christine Thompson showed that Shelby worked with species in Wacoita Bay, Massachusetts. But they suggested that the growth conditions could have an effect on the accuracy. They had accuracies in the 80% or so. because they were using those hatchery oysters, the larvae, to try to identify the field larvae. So the basic question was, would it work in the Chesapeake? And we published a paper in Limnology and Oceanography Methods that shows that it does work in the Chesapeake, but, well, I'll tell you that in a minute, sorry, keep jumping ahead. The work in this whole technique has a lot of work initially, because first you have to spawn and rear larvae, known larvae, or obtain them from hatcheries to create a library. And then you have to prepare these larvae, you have to image them all and create these libraries, which we call training sets. And then you use those to classify your unknown larvae. So the initial upfront setup is time consuming because you're trying to create your libraries. But Scott Gallagher and I are working on universal libraries that you can use all around the world. So we're going to add in laser spectroscopy, and hopefully that will help enhance the identification accuracies. So training sets are images of known larvae. So this is what you use to train the computer software to identify the unknowns. So this is just an example of a 3-species training set that has 250 images in each of the categories. And then you go out and sample unknown larvae, and you use your training set to identify what you sampled. So what we found was the differences in rearing temperatures resulted in differences in accuracy. So we can increase our accuracy up to 99% if we can account for the conditions that are out in the field. So meaning I basically reared these larvae in the same conditions that I sampled them from. So yeah, the accuracy increases from 67, 88 up to 97, 99%. You could match mostly temperature, but salinity was also an effect. So the way they form that shell likely has, the temperature and the salinity likely has something to do with the way they form the shell of the larvae. That's why these patterns might vary. But if you account for the differences, you can get high accuracies. So, as I jumped ahead before, the next step was to develop a fast way to image all these. It's still a lot faster than it used to be because you can see these birefringent bivalve larvae under the microscope, but it would still take you a long time to try to image all of them. So we developed this benchtop automated system to use with it, and we decreased our sample processing from 2 to 13 hours per sample down to 46 minutes a sample. And we found a lot of different quality control issues in terms of the light source settings, and so we were able to monitor all these. And hopefully this paper is going to be, this paper is in review right now at L&O Methods. But we set up tests and we set up validations. We showed that with the proper settings, this image acquisition system can yield quite high accuracies for identifying oyster larvae. And it's got applicability for other samples, so other types of organisms. So even if you don't use birefringents, we're essentially putting a Cedric raptor slide on an automated stage. We're imaging the entire slide. So for us, we take all those images and then I run them through a program at MATLAB that basically looks at the image and says, is there birefringents? If there is, it puts it in one folder. If there's not, it puts it in another. And we went a step further and tried to do what we call row cropping, which means it would automatically take that larvae out of the image and store it in a training center or in an image to be classified by a training center. So there are some issues with that, but we're still working on it. But anyway, getting the samples down to 46 minutes was a big challenge, but we did it. And some other researchers are seeing if they can use this with copepods. So once we were able to identify these oyster larvae and other types of larvae, but more importantly, focused on the oyster larvae, we went out in the field and sampled. And we tried to get at this question of behavior and mortality. So we want to know what factors cue the swimming behavior of the larvae. So as I said earlier, the larval stage is planktonic, so that's what this represents, these dots. The larval stage is the only time that P. virginica moves in terms of population. So oysters, the only time that their population actually physically moves is when they're larvae, or if we move them, if people move them. But once they attach to the shell, they're sessile. They're there forever. So they're epiphyleal, meaning that they need a hard substrate to attach to. So understanding that stage is really important to understanding the ecology of the species, to understanding the sinks and sources, population connectivities. If you put a reef there, is it going to produce larvae that will end up here? And in Chesapeake Bay, that's really important because if we can produce sanctuary reefs, or anywhere really, these reefs, if we create these particle tracking models, we can learn where the larvae are going to go, and the larvae may end up in other areas that are harvestable. So that way the sanctuary is supporting the fishery community, but it's also being reserved itself. And some of the larvae will be retained in that sanctuary also. So it's important to try to understand that question. So studies before have shown that larvae may be cued to swim vertical by salinity. So salinity may cue these larvae to swim. And again, as I said before, the vertical position of the larvae is very important, especially in an estuary like Chesapeake Bay, because if the larvae are at the surface, they're going to go one way, and if they're near the bottom, they're going to go a different way. And this just gets at what I was saying about the sanctuary. So in Maryland and Chesapeake Bay, we have areas in green that are sanctuaries, and other areas that are fishable. So the first thing that we did to try and assess this was to go out and see what conditions were like, to take a snapshot of the Chesapeake River. So we took this little boat out, this Parker, and we cast a CTD, so we were able to get different water parameters, temperature, dissolved oxygen, salinity, and we basically pumped water up into a plankton net. So we were able to get the exact parameter of salinity and temperature with the sample of larvae that we sampled. And we've collected 26 samples total, one at the surface and one at the bottom of these places, and we pumped 200 liters of water for each of these samples. So here's a picture of what the size classes of larvae look like and how they can be different. So these are oyster larvae, and this is less than 106 microns, which are called the D stage, because they look like D, the capital letter D under a microscope. And then there's 107 to 199 micron range. And then when they start to develop an umbo, which is this protrusion at the end, they're usually greater than 200 microns. So I'm defining these three size classes just by the way that they look on these fluorescent patterns. So we were able to make these abundance maps to see where these larvae were in the river before we were going to go out and do a larger cruise, which we're going to talk about. And we found smaller larvae up by this area, Broad Creek. And I don't know if you guys have heard of Harris Creek, but there's a lot of money, federal and state, that has gone into it, and that's right over here. This study was about, it was happening at the same time that they put some of that in there, but there were some plantings the year before. So I was able to look at the three different size classes and note the color change here because the scale is quite smaller for the larger larvae. There's not nearly as many of these greater than 200 micron larvae out there as there are smaller larvae, which is sort of expected for our selected species. You have high mortality rates there. But one thing to note is that there's different patches of the larvae. And this is likely due to the fact that these larvae are getting spawned from different areas of the river, meaning there isn't just one location of spawning that's happening throughout. And these are some of the first maps we were able to generate in terms of larval abundance in the world because we couldn't identify them before. So we kind of went out and did that survey just to get a snapshot of what conditions would be like for a more intense survey. And the more intense survey, we occupied 2 different stations. So we sat on the stations on a much bigger ship in July of 2012 when oysters we thought they would be spawning. We wanted to make sure there was larvae out there, and there was. And we used the RV Sharp, so a much bigger research vessel, and we were living on it. So we sat at one station for 2 days and just pumped water, and then we sat at another station for 2 days. So again, we did 200-liter samples from 5 different depth intervals every hour and a half, and I'll show you the schematic. We had a CTD, and we had a hose attached to that we pumped the samples up from, and we were able to get the exact parameters of the water, so salinity, temperature, chlorophyll, those things, and that way we could match that with the samples to see what kinds of relationships there were. And we also employed an acoustic Doppler current profiler, and that was to get at the current velocities to see what those were. So here's a schematic of the time series of sampling. So on the y-axis is the depth, and each of these dots represents the midpoint of where I sampled. And on the x-axis is time. So every space between the dots in the horizontal direction is an hour and a half. So this is the highest sampling resolution that anyone has ever done on any larvae. So I hope that we can do more, in different places. But this is, so the first thing I looked at was current with the acoustic Doppler profiler, acoustic Doppler current profiler. And I saw that you basically have an ebb and a flood tide, as you expected. And you have some pretty fast current, 45, that's meters per second. And with that tide, the flood tide, came more saline water. So this is all these axes are always going to be the depth. This is going to be that time, that hour and a half interval that I showed you, that sampling resolution from the previous slide. But what happened was during the flood tides, we had a lot of salt water coming into this first station that also had hypoxic water. So the Chesapeake Bay suffers from hypoxia in the summer, and we think what was happening here is upwelling was occurring. So we were getting this water from the deep bay coming into the smaller estuary, the sub-estuary, bringing this hypoxic salty water. So here's some schematics of the larvae. So 90% of all these larvae were present above this hypoxic, so the 2 milligram per liter hypoxic area for oxygen. All the larvae, most of the larvae were above that. And 89% of them were shallower than what I call the maximum salinity gradient. So the biggest change in salinity in the vertical direction. So the larvae seem to be getting cued by these two factors. And these are the larvae of the smaller size classes. And the dotted line depicts the salinity gradient. I'm sorry, the solid line depicts the salinity gradient, and the dotted line depicts the hypoxia line. So below that dotted line is hypoxic water. And again, the scales for the larger larvae are much smaller, 85 numbers per meter cubed versus the 4,000 of these smaller stage larvae. Station 1 was difficult because we didn't really have the tide was moving so fast that the larvae, if you notice, you can see, if I matched this up with the current, you'd see that all these larvae were in that ebb flow water. So they just sort of sloshed back and forth, and I didn't get to really tease out any kind of behavior from this. But it's still interesting because they still stayed above these, hypoxic line. What's that?
Diurnal pattern?
Oh, of tides? Oh, no, there was no diurnal pattern. So I looked at that to see if they migrated up or down with the light. They did, yeah, I looked at that. So the second station was further up in the estuary, and we didn't have this upwelling event happening. The currents were a lot, they weren't quite as high, and we were able to get sort of a better depiction of a salinity gradient. It was a lot shallower in the water column as well. So, and these are warm temperatures, so they're spawning at this time. We still had hypoxia, but it wasn't nearly as prevalent as it was in Station 1. So here you'll notice the larvae, they're more ubiquitous across. So they're not only in one tidal cycle. They weren't just at that time, they were ubiquitous across both. But you still see they stay above this maximum salinity gradient, these smaller stages. Now the larger stages, again, the scale is going to be a lot lower for these large larvae because there aren't as many. These larger larvae start to penetrate below this maximum salinity gradient. So this indicates behavior, or could indicate behavior. They're actually sinking down below when they get larger.
What is the maximum salinity?
At Station 1, the maximum salinity, the actual salinity or the gradient are both. The salinities here were between 13 and 16. So station 1 had higher salinities because that water was coming from the deep bay, and the salinity gradient between that water and the estuary water, the chop tank water, was around 1.5. That's how sharp that line had in terms of the change. So that's what cued the larvae to swim. My advisor, Dr. Elizabeth North, has a particle tracking model that she estimated what salinity should cue the larvae to swim before we did the study, and based on some other things, and she put 1 in as a factor. And so what we found, though, this salinity gradient was a lot smaller, so maybe 0.5 or so. But what we found is 1.2 is where all these smaller larvae stay above 1.2 in terms of salinity gradient. So we figured out that salinity appears to cue their behavior. Oxygen may also, but we need more studies to see if oxygen does. It's kind of hard to tease those two things out, but it likely does. There's laboratory studies that show that too. And I didn't find a lot of them in the hypothesis. I mean, 89% of them weren't, you know. So the next thing we wanted to do was calculate mortality. If you can calculate mortality, you can create not only larval tracking models that will predict where they'll go, but you can try to estimate the mortality of those larval populations. And in the end, you want a holistic view of your oyster population or scallop or mussel or whatever species you're trying to restore, just for basic ecology. So in traditional fisheries, they use what's called a catch curve approach for calculating mortality. And there's some assumptions with it about that the population has to be a closed population, constant mortality. Your sampling gear has to catch all the different stages of the same amount. And basically you take this slope of a log length and you plot it against age. And this is by Rutgers, so this is pretty traditional 1970s way of calculating mortality in larval fish. But no one tried to apply it to invertebrate larvae. So I did that, and then I also used something called a vertical life table approach, which a lot of folks that study copepods use. So essentially I just used 2 different techniques to calculate mortality so that I could compare them to see if my estimates were close. So for the catch curve, I was able to figure out, we reared larvae, we were able to get a length-age relationship. So I identified about 86,000 larvae in those samples for that big station cruise. And I have an actual measurement for every one of those because the software program, we programmed it to measure the length of those shells too. So by having a length for every single one of those larvae, those 86,000 larvae, I was able to ascribe an age with this relationship. So then I had an age, and then I was able to use these mortality techniques to estimate mortality. That mapping cruise where we went all over the river, and then the 2 stations of the fixed cruise. I told you there were some assumptions. So we violated a few of the assumptions. The most accurate, and I talk about this in the paper that's getting submitted soon, the most accurate estimates are from station 2. And this is because at station 1, we had a lot of advection happening with that tide going in and out. In station 2, it was ubiquitous. We didn't have a lot of movement, so it didn't violate as many of these. The coolest thing for me about this was that the two methods yielded very similar results. These error bars are the 95% confidence intervals with the catch curve approach, because the catch curve approach uses the continuous data set, so it uses all the data. Vertical life table approach just uses 2 age structures, so it doesn't use all the data. But it's interesting how both methods were coming up with similar values. So these are some of the ranges. Oh, I just spoiled it. Spoiler alert already. So sorry. So this is why the calculations at station 2 really have. Because at station 1, we had a lot of displacement of water versus at station 2. So this shows that graphically. This is the water column direction, so I don't expect you to be able to read it, but this is just the depth. And you can just see that a lot more water is moved in different directions at station 1 and 2. Yeah. And this obviously means it's not a closed population because we have 2 different cohorts. So the mapping crease wasn't as accurate either. So in conclusion, this automated image acquisition system decreased acquisition time from 430 days down to 43 days. So it allowed me to get a PhD in a relevant amount of time. We were able to measure and identify these 86,000 larvae and get pretty high classification accuracies overall. And by analyzing those field samples, we were able to figure out that salinity is the dominant hue, which was thought to be, with 90% of all these size classes above the gradient of 1.0, and the smaller size classes about 100% or above the 1.2 mark. So we can parameterize the models with this. And then we were able to establish mortality rates. And as I said, the 0.37 was probably the most accurate larval mortality rate. And currently, Elizabeth North has a paper going into the Chesapeake model and symposium that uses larval mortality to see that value for that. So the impacts of this research are that these techniques can be used to enhance image acquisition for other plankton species, like copepods or pteropods. Those are food sources for salmon, those biofilms also. And this will help us understand the larval ecology of the oysters and their mortality. And it helps us provide a better idea of the conditions that cue their behavior. And it helps us identify the sources of things that I keep talking about for management areas, so that we can validate these larval transport models like this one on the Chesapeake River. And that will have applications for creating marine protected areas. So that just reiterates that for our management goals. Understanding the larvae can help us with long-term goals of setting aside certain habitat because it might enhance other areas. So right now I'm working with Scott Gallagher to try to add spectroscopy to this polarized light technique, because we want to be able to create universal libraries, so that a lab here can use the same training set library that the lab in Chesapeake or Virginia uses. So that's why I'm going to Woods Hole in April and then all fall. And I'm also working with Daphne Monroe at the Haston Shellfish Research Lab to create a connectivity, or create a larval transport model actually for Barnegat Bay, for hard plankton. So we're going to be able to identify the larvae in the field and try to sort of validate that model like I did with oyster. And this is another project that I have in the works to target living shorelines with marsh mussel larvae. So there's a marsh mussel larvae that called Buchensia demysa, and it basically, its visual threads form right at the roots of certain marsh grasses, and there's a synergistic relationship. So they actually help keep the marsh stable from sea level rise and certain other tidal factors. So understanding where larvae are out in the system may help different organizations put out living shorelines that could help catch these mussel larvae and help stabilize the shoreline. So it's identifying the larvae of those has applicability for shoreline restoration as well. So that's it. I'll take any questions you guys have.
Did you say you're looking for other countries to study?
I'm in 3 now. I'm in 3 now. Yeah, I'm always,
And can I just
Can you repeat the question? So,
Oh, yeah.
Yeah.
Move on, but.
Okay.
I have additional data. You said you're working with somebody at Woods Hole, Daphne Monroe, the Barnegat Bay?
Sorry, Daphne Monroe's at the Haston Shellfish Research Laboratory, and I'm working with her on Barnegat Bay.
Barnegat Bay, okay.
And then I'm working on a different project just to try to enhance the identification techniques with Scott Gallagher, Dr. Scott Gallagher at Woods Hole.
And are you familiar with the oyster restoration work that the American Little Society is doing in Barnegat Bay?
I'm not, no. The question was, am I familiar with the oyster restoration work that the Littoral Society is doing in Barnegat Bay? I'm not, no.
And also in the Delaware Bay?
I know a little more about the Delaware Bay because I went to their oyster stock assessment meeting. I sat with Dave Bouchek and Daphne. Dave and Daphne would know those systems better. I'm new with the system, so right now I'm trying to parameterize our larval transport model for clams. So I'm sitting with like a hard clam book this thick and all these other papers on heart clam, and I'm trying to land that species.
Okay, thank you.
Yeah.
I'm Greg Rivara from Cornell University, Quantum Extension. I want to say really great work. That's amazing. This is really good stuff. A quick anecdote and a good question. Going back to refall, what happened with refall, with the oysters set on them, I had a project shooting in New York in 2010. We had 12 small refalls, only about a foot and a half. That's good. We put them in a tank out in Southhold on Long Island and brought them to Jamaica Bay and took them to Garrison Creek and put them down with divers. Actually, I know you know Mutt Merritt pretty well. He's a good friend of mine, and I called him up before we even started, before we even had the refall, and what would you do in my case? And he said, why would you want to set oysters on refalls? Put them out in the bay. And they said, why do people do that? So, well, Jamaica Bay, oysters are fucking mistake. So fast forward to 2015, they're in the water 5 years. Most of the oysters didn't get eaten or crushed by a boat, fell off the reef wall. They got so big, I'm talking about like 4 or 5 inch oysters now. They couldn't sustain it, concrete fall, whatever happened. Just anecdote, not in the
Yeah, so if you're not getting, so I have a question about, what's the larval population in Jamaica Bay? So you're probably not getting any natural development, so those oysters just get big, and then they die off over time, sure.
I studied it behind me.
Yeah.
I would say close to zero. The question, though, with your work and looking at where these larvae go, it's all well and good to study a system, it's a nice sunny day in July. What happens 5 years from now? It's a big storm, and there's a big wind event, and the larvae just go elsewhere. I mean,
yeah, that's right.
I mean, it's not foolproof, right?
No, you're absolutely right. So this is a snapshot in time for these mortality calculations and in terms of the behavior, I think we're onto something with that value. And I'll challenge anybody to find another mortality calculation of an oyster larvae. There's Trinidad Stallworthy in Canada from like 1970s, and then there's a couple others, but not in this. So this is a snapshot, but it's the first look we're getting at that. So yeah, absolutely. Yeah.
What was your assessment of your larval mortality result? Did you say that was a high mortality rate or a low mortality rate? How did you feel about?
Well, the question is, what was my assessment of the mortality rate? So I had a lot of people on that was my dissertation work, and I had a lot of very, very smart people on my committee, luckily. There was Mike Wilberg, I don't know if you guys know who he is, but he thought that those were higher than what he would expect. But those were observed values, so I think those are the best values we have of larval mortality. I'm not surprised by them because it's an R-selected species, so we expect high mortality. Other invertebrate larvae, so 0.37, say, instantaneous daily mortality, is the value that I would stand by. And so Rummerl has a paper from 1990, he talks about sort of all these invertebrate larvae and says it's 0.2. So, and I'd have to draw it's an equation that you have to put that value in to actually calculate it. But it doesn't translate into percent directly. So it's exponentially higher than that. It's high.
Who wrote your recognition software?
Yeah, no, Scott Gallagher, I give him the credit for that. We did a lot of improvement on the software. So Scott came up with the idea with Sanjay Talari, and then him and his student worked on it. We bought the software from him, and then we made a lot of improvements to it to make it more accurate. Right now, if you want to get the software, you need to go to coastaloceanvision.com. That's how you get that. It's going to come with a system called LIDAT, Larval Identification Hydrologic Data Telemetry Unit that Scott created. So that's the way. We bought a copy from Scott that we were using for our own purposes, but now if you want to get the software, go to coastaloceanvision.com.
Jake, you mentioned the accuracy of the polarized light technique was sensitive to temperature and salinity. Does that mean once you have that training set the following year, if you have a different, if they're spawning at a different temperature time or there's something that's changed in the system, your set would be less accurate?
We found that the hatchery conditions were a lot. Basically, the hatchery conditions were actually a little bit, at least in the chop tank, were a little bit cooler than what we were finding out in the field. And we found, they obviously, the feeding's different, so phytoplankton community is different than they are in the hatchery. The biggest differences, though, in accuracy is with the 20% were if we reared them in really cool temperatures, so like 22 degrees Celsius instead of 26. So what I would expect that if you rear larvae between 25 and 29 degrees, you'll be able to capture conditions that are likely to spawn in Chesapeake Bay, and I would go a little cooler for New York.
So just once you've done that, though, you don't need to revisit that.
No, so the whole idea is to try to create these universal libraries, you know, that have a whole, that are made up of a whole range of conditions, and use this. We got to bring this, probably this spectroscopy in with that too, to make it applicable to all the different systems. But that's what Scott and I are going to work on. I have a really big larval collection that Richard Lutz had with over 1,000 different vials of larvae of species from all over the world, and I'm going to take those with me to Witzel and see what I can do in terms of making a universal library, and that's the next goal, you know.
Great, thanks.
Could you elaborate a little bit more on the salinity Q? You seem to say that the gradient, which I came away with the gradient being maybe more important than the actual salinity value. So that I may be missing.
Yeah, that's right.
Yeah, but so what's happening? I mean, are there sensing changes, and are they going up, are they going down, or is it?
So what's really happening, I think, is that they're sensing change. So one thing I didn't mention is that these larval shells are negatively buoyant. So if the larvae wasn't swimming, they would sink, right? So the larvae are hitting the salinity gradient, a change. They're sensing that, and they're swimming up. There's some preliminary lab data that shows that, but the lab data couldn't get it, the gradients that we experience in the field.
Going upward,
they're swimming. They're going to swim up.
If the salinity goes up or down, are they still going to swim up, sensing the gradient?
They'll swim up. So when the flood tide comes in and the salinity gets higher, they've been shown to swim upwards. That's Mel Carricker's work from before.
But when it goes down?
When it goes down, they're still found. I mean, the smaller species are still found high in the water column. The larger larvae, I would guess, would go below, but they're going below these big gradients anyway. So I think the small larvae would just swim upward. I mean, that's what I saw. That's what I observed.
I'm asking about this modeling, so larvae. So first question is, how is the status of modeling on this larvae tracking? The second one is, what's the major problem? What's the major difficulty in tracking?
Okay. So what the status is, right now? There's a larval transport model. It's open source. You can Google that, and it'll come up, and you can apply it to different systems. That's what we're going to apply to Barnegat Bay, this larval transport model. And we couple that with a ROMS model, so a regional oceanographic modeling system. And the ROMS model utilizes Navier-Stokes equations to help us understand the natural flow of the water. So it's pretty accurate, mainly at University of Maryland, Hong Kong.
So consider the weight of the lava, or just like
So the ROMS model helps identify just sort of the water flow. Then you add a behavior component to the larvae. So when a larvae experience that salinity gradient in the model, it'll cue them to swim up when they're a certain size. But when they're greater than 200, they'll go below that. So you can pinpoint their behavior in the model. And then with the mortality, you get into more future cache, and rather than space, you know, where the larva is going, you're getting into more time dimension of trying to figure out your population and how many of those survive.
So the tracking technique is already quite good when you put this in.
Yeah, it's partial tracking in a model, so we're not tracking them in the field though. But yeah, it's the validations for the models, at least Elizabeth Chaperon's model, have shown pretty good results. I can give you the paper in 2008 on that.
Hi, I'm Jenny Marshall from the City of New York, and I'm new here because there's been a revolution in technology. I work with conservation genetics, and now we can sequence DNA from water and from soil. When you were, I think I talked to you a little bit earlier, when you were talking about the reef balls and the control site right next to it, and then another control site, that's more or less from my students right over there. That's what we're working on. Alison Fitzgerald found you in the Bronx. And we just started, and we're validating the method with morphology. Marina's doing a lot of that. And I just wondered, like, it seems like it's a very rapid method once it gets ground truth. Would it be useful to apply this more broadly to assess the biodiversity recovery of the reefs and the nearby areas?
Yeah, that DNA tool to see what in the sediments. I think so. I bet you'd get different signatures in the, you know, if you went underneath a restored reef versus outside. Absolutely. It's a very good tool.
Sort of similar to that, I was wondering, and I know you sort of had a focused question with the reef balls, but I was curious if there was any other research that you there do or looked at with trying to understand how reef balls compare to a reef as a potential control, or where in the spectrum?
In terms of like just
biodiversity and things like that.
Of what kind of reef? Of just an oyster reef versus the reef ball?
Yes.
Well, essentially in Chesapeake Bay, most of our oyster reefs are just those spat on shell areas. So there aren't really. Before I went to get the PhD, I worked in campaigners lab. So for like 10 years, I dove on all these different sites, and you know, I would dive down, and mostly after 4 years, we had a lot of poaching would happen in the sites. But really, it's really just a lot of the areas are just flat areas. So I haven't done any work on comparing reef balls per se to an area that I know is an older sanctuary reef. And I don't think I haven't found anything in the literature on just how well oysters grow on the reef balls. So this was just a preliminary. This was not a lot of money. It was just a quick preliminary study just to see if the oysters grow on the reef balls. So to help, it's really Chesapeake Bay Foundation is really trying to get at that question. Does that answer that?
Yeah.
Okay.
This question on the reef balls, you had size over time after 4 years. Did you differentiate whether the shells you measured were on a live oyster or not?
No, those were just all the live oysters that the diver took out of the fly jack and brought back.
So if they're attached on the reef ball, did you measure it? Did you bring up the reef ball to measure it, or?
No, they chiseled all these oysters off and brought them up. Okay. We took them back to the laboratory.
But did you only then measure the live oysters, or just at a shell? That was a certain thing.
We measured boxes, but there wasn't a lot of mortality. So we called that a box is an oyster that had died out and left its shell. It wasn't significant. I mean, there was maybe a couple of boxes in all of them. I mean, the survival of oysters in all these areas seemed to be good.
So disease is not that prevalent in this area, or?
Disease there was a prevalence of derma, but it's not. It's a mesohaline region, so it's not like the high salinity regions in the south, Virginia. So there is derma disease there, but I don't think it's affecting mortality that high. But as we talked about, conditions change. We get a drought year and the salinity rises. I would expect the prevalence of Pictus marinus to increase in the populations of oysters.
Question one: if you put a field of these balls, they're permanent.
Yeah, once you put them out. Yeah, fishermen love them because they attract fish. So actually, for me to drive, I was driving that boat, and for me to get to the site with the divers, I had to kindly ask the recreational fishermen guys if I could go to the site because they want to fish there, and they're all over the site.
A regular fishing boat, oyster dredge boat, they never move them.
It would move them, but it would be a really big pain. It would cause them a lot of havoc. It'd probably rip their gear up a little bit. So they wouldn't want it.
And weigh about 300 pounds.
Yeah, I mean, once they got all the oysters on them. And so if you put those in sanctuaries, and this is why, I mean, you know, off the record, why the Chesapeake Bay Foundation was putting them out, they can just sort of put it in. So, and if they enhance the populations of reefs and those organisms, those mussels and all those other organisms that grow on reef balls help with denitrification, and they help filter the water also and create habitat for bigger fish that come into these fishermen fish. So I like them personally. In my own opinion, you know, away from the study, is I like their reef balls.
Was there a progression of settlement on the reef balls, mussels, and this and that competition for the oysters?
You know, that's a really good question. So the oysters were preset on these reef balls. So if you just put a reef ball out, say in high salinity area in Virginia, where you're going to have high recruitment, that's the question, you know, are you going to get this recruitment from these oysters, or are these other species going to outcompete the oysters to settle on those reef balls? I don't know the answer to that. You know, in the mesohaline region of Chesapeake Bay, putting these out with oysters already settling appears to be a good way to enhance the population of oysters. And I can tell you after 4 years, you know, after 4 years, the oysters survived on these reef balls. And I do have initial numbers for how many oysters were on the reef balls. I just didn't want to throw the number up because I don't have it for the spout and shell. But I can tell you that the oysters on the reef ball survived after 4 years. And the site at the north that I didn't talk about here, the reef balls were put out 2 years later in the spout and shell treatments, and they still had more reef-associated fauna, still had higher abundance. And so the pattern, you know, continues, but there's a lot with that, so it's better to read about that in a paper than for me to try to present it, because it's just a lot of different studies. But yeah, even after 2 years, they recruit a lot more than areas around in terms of the associated fauna.
The reef balls, do they have a higher amount of oxygen in the water? That's what attracts the fish there?
I don't know. I don't know. Yeah. They have, they've got, I think I would speculate that the reef balls, because of the three-dimensionality structure, they have these holes inside of them, and because of the, all these smaller fish that live on, so I didn't talk about the smaller fish because they swim out of the way when the divers trying to sample, so we don't know how many blennies and gobies were on, you know, the reef balls versus not, but they saw them, the divers saw them, the divers saw the fish swimming around the reef balls. So I think the fish are attracted to food items on the reef balls, and they're attracted to the actual structure. And it could be actually, I don't know.
Quick question about the initial channels on the reef balls. Did you look just on the outside or the inside of the ball?
We only looked on the outside. The diver actually couldn't even. So that comparison is an underestimate of the actual population because we couldn't even look on the inside of the reef ball.
Hi, I'm Melissa from the River Project. Just a question about shell meat. You touched upon it a little, but what are the benefits of shell meat versus PCR or fish? Because it seems like those would avoid the problems like changes in translation.
Yeah, there's some contamination issues when you try to create markers. So we did a study. Well, Matt Hare from Cornell was on Christine's dissertation committee for that project, and Matt actually contacted me to try to. He was talking to me about verifying his markers using this technique. He does a lot of the PCR work. There's contamination issues with DNA, and we did a blind study, and we sent some off to IMET, to our place at IMET, some samples. Somebody created them, and then they gave me some. And IMET came back with species that were from the Pacific Ocean. So I think that the genetic is going to take this over. It's going to surpass this technique. So it's just not there yet. You have to create a lot of markers ahead of time. And as I recall writing the paper in limnology and oceanography, there's some step in the beginning. I don't work with DNA, so I don't know exactly. There's some step in the beginning where there's contamination issues. So you actually get, if you look at Christine's paper, you get higher accuracies with this technique than you do with PCR right now.
Couldn't it be incomplete database too, that it just assigns it to, well, this is a specific possession in the database?
Could be, yeah. So I think that's part of the limitation is they don't have all the species put in yet. So you can't just take a jar of water and say, hey, what is this? You know, not yet. So, but I think it's going to surpass, you know, eventually. But this technique can tell us a lot right now, and it can tell us a lot about certain shell formation issues and ocean acidification. So, can I bring this up again? I think I have a slide at the end. So another interesting thing that I should have probably told you. Oh, try this, yeah. This one. So that slide here, the next slide. So, no worries. Well, so you can see the difference in the shell pattern from different pHs. So ocean acidification, we might be able to detect that. I'm talking to Scott Gallagher and Nina Zurich from the West Coast, and we're going to talk about looking at pteropods in Antarctica to see if we can detect this, we're going to propose, right? But we're going to see if we can detect these changes. So that's another reason that this method might be good in the future. Like I said, real-time PCRs are overtake this in terms of identifying, certain things. But this is ready to go right now. So those were. Sorry. One more time. All right. So the top panel is oyster shell that was in a pretty acidic solution. I did this with other solutions too. That's not going to, 4 is not going to be characteristic of what's in this environment, obviously. But, and that was a month. But you basically can see this happening even in low pHs like 7 and 6. The differences. So if you keep the sample buffered, you retain these. I'm scared to press the button. You retain these patterns like you see on the bottom. And with acidified conditions, even at 7 pHs, you start to get deterioration. So,
but how does that storing a dead shell, even at 7 or 8? How does that compare with the live animal growing?
Exactly.
I mean,
that's a very good question, and so that's why this is like a preliminary, like at the end of the talk. I don't know, and the reason I wanted to do this was if I encounter patterns like that in the field, I want to know what the difference is. So if you want to do a study like this with, detect acidification somewhere, I mean, an oyster larvae, these larvae organisms live in all kinds of pH, right? So that's probably one that's not going to be affected as much, because they're already able to live in these, conditions that are range between, high 6s and 8s. But other species like pteropods remain. So you want to set up controlled experiments. But it's just another direction. I mean, it seems to detect these differences in cell structure.
I think we've exhausted you, Jake. We've got a quiet group on the webinar that has questions. But just a reminder, our next seminar is a Tuesday, by the way, but April 26th. We're going to hear from Tim McKenna and Frank Nishi from Columbia. They're going to talk about, again, we're going to go back and look at Irene and Lee and the effect of the sediments that came into the river at that point.